Hello, my name is Ryan Scott. I'm the project scientist for the NASA Ames Life Sciences Data Archive, and I'm thrilled to be speaking to you today here at From Images to Knowledge 2022. When we're here, because when you think about going towards deep space, there's a real need to understand the fundamental mechanisms and develop different health systems to counteract various spaceflight stressors and hazards. Here they are listed. And to really understand these stressors and hazards and develop effective countermeasures, there's various different platforms in sort of a progressive manner to really build and build deeper, so that we, from ground experiments to low Earth orbit to lunar missions, that we can identify the fundamental biology. We can then build the systems and the tools to support ultimately human life, as well as crops, as well as a thriving ecosystem, and eventually have this foundational experiments through lunar science, so that we can sustain life on Mars. And there's been some really exciting goings on in the past couple of years as we move towards the Artemis missions for lunar science biology. And this is a schematic image that shows some of the planned experiments and the various different model organisms that are going to be part of proposed labs and lunar surface payloads to conduct experiments and collect data from the moon, from lunar experiments. This is a report, the QR code there. You just take a screenshot with your phone, and you'll be able to go to a lunar biology report that was recently produced. And this is a little bit of a thinking of the full scope of the model organisms, from simple organisms to more complex, that translate for in different parts towards human health, that are part of this entire space biological domain. And NASA on its whole is very, very dedicated to open source science. And this is very exciting, that from various different policy changes to new solicitations encouraging open science, open software elements, supporting core services for high end compute, and through all these different options, really develop a community building effort to form partnerships that open science can be performed to be maximally inclusive and have maximal access for all, for greater diversity in the scientific field. And this is a link to the main website, and this is on the level of headquarters at NASA that is leading and believes in this as a philosophy. When it comes to open science for life in space, this tagline above me, think about all the various different experiments, from ground radiation experiments, ISS experiments, future lunar experiments, and others, that take these various different model organisms that we deal with in our group, and these data through the various PI experiments, and after they finish publishing, they take their data and they submit it into a submission portal. Various different biospecimens, though, that come from those experiments are either sequenced by the GeneLab sample processing lab, or stored in the biorepository known as NDISC, shown here. And eventually, all this data is brought into our OSDR, the Open Science Data Repositories, made up of ALSDA, but also NASA GeneLab. And when users access NASA GeneLab and the OSDR, there's a collaborative workspace for data sharing and for upload. There's also a well-established omics analysis toolshed based on the Galaxy platform, and various different pipelines that have been decided by our user analysis working group community. And this has led to tremendous knowledge gained. If anyone's interested, this is a QR code to a package of Cell Press articles that were published in 2020. In majority, were largely based on GeneLab data access itself. Beyond that, the repository itself, when you take the data, there's a number of different downstream analytical tools that are being provided for. One is an environmental analysis portal to understand the various confounds, from radiation, O2, CO2, relative humidity, temperature, those types of environmental data. There's also the data visualization, which is taking the results from the various assays and looking at them in different forms to do analysis across missions. And lastly, the new one is an AI machine learning knowledge graph modeling portal that's underway right now, that we're very excited about. And when it comes to bioimages specifically, I thought I would take a moment or two and review a little bit of the full scope of what ALSDA is dealing with bioimages. First of all, the data is very multi-hierarchical. from cell to organ to tissue to the whole behavior and the ecosystem. Multimodal, you can see a list of the various different modalities of the bioimage data and structures that we're developing standards for, and very heterogeneous when it comes between missions and strains of animals, durations, ages of animals, and other different organisms. And these are just some snapshots when you think about on the, especially that multi-hierarchical level. This is shots from bone. There also is here's some fluorescence microscopy images from the retina, looking at a space-flown mice retinal changes, as well as light microscopy and micro CT again, just to give you a little flavor of some of the bioimages that we're collecting and curating. Beyond small animals, rodents, there's also microbial imaging and various different plant imaging to be considered, as well as crop growth impacts to be considered for why bioimaging is so important for spaceflight biology. This is a picture of a gravitropism on how roots are affected by gravity. So something to consider. It's really important, that little bit I put in there on standards, because standards are going to be key to ensure reusability of data. And in the last year, once NASA GeneLab and ALSDA integrated, a whole group of at least 100 different scientists have rallied around ALSDA and helped us help by providing feedback on the scientific standardization of our metadata, as well as sort of ideas on how it can get better reused to ensure reusability, reproducibility. And we are going to be moving more towards bioimaging, and we would love to have any more SMEs join us. There's the email to join if you'd like. And this is just a snapshot of the various different assay, what we call configurations. It's based on the ESA tab model for investigation study assay. And in the past several months, just several meetings, we've developed about 17 of these new configurations for metadata, so that as data is submitted, it's curated with strong and rich metadata, so that there's good provenance and a good ability to do cross comparison using the same assay across various missions and studies. So we've also been producing pilot projects to demonstrate reusability of data. Later on in this presentation, Lauren Sanders will be presenting more on some microscopy data, looking at deep learning, but there's also benchmark efforts that are going on. And there's also several other different pilot projects, just as not necessarily on bioimaging, but on how do we take all these data that have been richly curated and structured so that they can be brought towards transfer learning, but also causal inference, federated learning, also just cross-comparison meta-analysis, as well as using knowledge graphs and directed acyclical graphs to look at risk probability for health hazards for space. On the future, that AI ML knowledge graph modeling portal, we're really excited. And just to give you a sense of how we're looking at this, GeneLab has raw omics data that through their AWGs have established a very standardized omics processing pipeline. For ALSDA, you can think of the tabular results that we have from our experiments, but also then the images that we're working and looking at computer vision as well as transformer models to take these images, produce information then that can be mined and then conjoined with the omics level and other different data, so that you can have this sort of multi-hierarchical data brought into one and eventually applied and utilized by our modeling zoo, be that a number of different modeling zoo, ML, AI, knowledge graph tools that we're looking at for our analysis portal. So it's something that's happening real time, and we're all really excited about it. So the big picture for open science for life in space, we have our submission portal and an analytical portal, sample processing unit, the work that we're working on towards imagery as well as video and other different applications. And through that, to enable access for the greater global community to mine this data, to have new knowledge at a faster pace, be more inclusive and have access. And of course, this does depend on a firm foundation of our data being fair and maximally open access. And thank you so much for today. I do need to thank my colleagues. Absolutely. Everyone here knows that science is inherently collaborative between Ames Life Sciences Data Archive, GeneLab, our AI/ML team, the biorepository at NBISC, as well as our biospecimen sharing program. It's all one team effort. It's an amazing collaboration between teams, as well as our amazing AWG members, the various collaborators, and of course, thanks to our stakeholders that believe in open science for life in space, NASA Space Biology, the Biological Physical Sciences, and the Human Research Program. So thank you so very much, and I'll take any questions.